Hello students, once again welcome in biology class. In this video, we are going to study about the enzymes involved in the DNA replication process. This is the second part of our topic DNA replication. In this video, we will study that what are the enzymes which play their role during the process of DNA replication, and also what are the roles of those enzymes. This is one of the very important part of DNA replication process. As we know, DNA replication is a very important multi-step complicated enzymatic or complex enzymatic process. So without the help of these enzymes, this DNA replication process cannot be possible. So that's why whether it is initiation of DNA replication or the polymerization of nucleotide or the joining of the nucleotides, enzymes are required in every step of DNA replication process. As we know that DNA is a double helical structure, and DNA replication start by the process of unwinding of those two stranded, and after unwinding of the two parental strands, the new synthesized strand are formed. So these are the 5 important enzymes which play their role in the DNA replication process, and these are the function which are performed by these enzymes. This is also important in the respect of board exams, because many times it is asked in the board exam that what is DNA replication, mention any 3 or any 5 enzymes with their function. So these are the 5 important enzymes which play the role during the DNA replication process. First one is DNA polymerase. The role of DNA polymerase is also known as DNA dependent DNA polymerase. So DNA polymerase or DNA dependent DNA polymerase. The role or the function, it polymerizes the nucleotides or polymerization of nucleotides. As we know, DNA is made up of nucleotides, the units which combine together and form the DNA, that is nucleotide. So the DNA dependent DNA polymerase enzyme helps in the polymerization of nucleotides. Like if we assume that this is the unwinded helix and this is the 3' 5' polarity strand, and this is 5' to 3' polarity strand. So here the continuous synthesis takes place, and on this strand the Okazaki fragments discontinuous synthesis takes place. So here these are the nucleotides, and thus the nucleotide polymerization means their chain formation of the chain is done by the help of DNA polymerase enzyme. So the number 1 enzyme, DNA polymerase, and the function of DNA dependent DNA polymerase is polymerization of nucleotide. Another important enzyme is helicase. Helicase, its function unwinding of DNA strands. As we know, the DNA replication starts with the unwinding of double helix. As the DNA is a double helical structure, this is the structure of DNA. So there are specific places which are called ORI, origin of replication, from where replication starts. So if we assume that ORI is here and it unwinding starts from here, so by the help of helicase enzyme, this strand starts open. These strands are become opened like this. So helicase enzymes helps in unwinding of DNA strand. So the second enzyme is helicase. It helps in the unwinding of DNA helicase. Third is HD protein, or it is also known as SSB protein, single strand binding protein, or HD protein. The role of this enzyme, it protects stabilization of unwinded strand. As we know, the unwinding is important for the DNA replication, and if it remain unwinded after some time, it can re-stabilized like this. If these are the unwinded helices, these can be stabilized again. So to remain these unwinded helices unwinded, HD protein. So HD protein helps to maintain the unwinding of the unwinded strands. So HD or SSBP protein protects stabilization of unwinded strand, because if these strands again gets unwinded, the DNA replication will not takes place. So unwinded remains by the help of HD protein or SSBP protein. The another very important or fourth enzyme which is very important, this is topoisomerase. As we know, whenever the uncoiling takes place or unwinding takes place, the tension is increased in the another end. Like if this is the unwinding, so in this end, this kind of tension is increased. As the unwinding takes place, another end of DNA replication, the tension is increased, and because of this tension, the further unwinding can become difficult. So to release this tension, to release here the tension is generated because of this uncoiling. So to release that tension, topoisomerase enzymes play its role. So release of tension at on end of strands. As unwinding takes place from another end towards the another end or towards the both the remaining ends, the unwinding takes place. The tension takes place, and this tension is released by the enzyme topoisomerase. So topoisomerase helps to release tension. DNA ligase is another very important enzyme, and the role of this enzyme is it helps in joining of Okazaki fragments. As we know, if we consider that this is the 3' 5' template strand, and this is the 5' 3' template strand. Here the polymerization or replication is continuous, but here it is not continuous, it is discontinuous. These are the small stretches of nucleotides, and these are arranged by the RNA primers. So here this strand is not continuous. These are the Okazaki fragments. These small nucleotide sequences are called Okazaki fragments. These Okazaki fragments need to join. Without their joining, this lagging strand cannot be completed. So the DNA ligase enzyme helps to join these Okazaki fragment, and it becomes a single strand. So the DNA ligase helps in joining of the Okazaki fragments, and these Okazaki fragments are the part of lagging strand, and after the joining, these Okazaki fragments forms the lagging strand. In this way, we can conclude that without the help of these enzymes, this DNA replication cannot be possible. So to complete the process of DNA replication, these all enzymes are very much important. So let's summarize. These are the 5 important enzymes, and these are the functions. DNA dependent DNA polymerase, polymerization of nucleotide; helicase, unwinding of DNA strand; HD protein or SSBP, single strand binding protein, it helps or it protects stabilization of unwinded strands. Topoisomerase, it is another important enzyme, and its function is to release of tension at on the end of strands. And the DNA ligase is the 5th important enzyme, and the function of DNA ligase is the joining of Okazaki fragments. So this is the end of this video. I hope you understand all the functions of all the enzymes which are involved in the DNA replication. I will come in front of you with new topic. Till then, wait, watch all the videos carefully. If you have any query regarding it, you can comment to the video, and you can also send your queries directly through the WhatsApp to my personal WhatsApp number. Till then, thank you for watching.